Hello everyone. Our tutorial is on cardiac muscle and the conduction system. So welcome to the tutorial on cardiac muscle and conduction system. It's one of the most fascinating and high-yield topics in cardiovascular physiology. In this session, we'll explore how the heart achieves two extraordinary feats. First, how it contracts powerfully and rhythmically, and second, how it does so in perfect coordination through its electrical conduction network. But this tutorial isn't just about learning the facts. It's about learning how to answer the exam question. You'll see exactly how to think through the topic, how to structure your essay, and how to present your ideas clearly and logically for maximum marks. We'll start by discussing what the examiner expects, then move through the key sections, show how each part connects to the heart's role as a synchronized pump. As you follow along, ask yourself the following questions. Why does each feature exist? What purpose does it serve? How does it maintain cardiac efficiency? That's the level of understanding that transforms a good answer into an excellent one. So let's begin. Before we begin, here's what you should be able to do by the end of this session. First, identify the unique structural features that make cardiac muscles special, the very design that lets it beat continuously. Second, explain the ionic events behind cardiac action potentials, how sodium, potassium, and calcium shape each phase. Third, trace the normal conduction pathway from the SA node all the way through the Purkinje fibers. Fourth, understand excitation contraction coupling. That's how an impulse becomes a mechanical heartbeat. And finally, relate these electrical activities to ECG changes, connecting physiology to what you see on paper or screen. In short, by the end of this class, you won't just know how the heart works. You'll understand how every beat tells its story through structure, ions, and rhythm. So now that you know what to approach or how to approach the topic, let's lay the foundation. What really makes the heart such a perfectly coordinated pump? The heart's rhythmic contraction is what maintains continuous blood circulation throughout the body. Every beat must be strong enough to propel blood, yet precisely timed to refill before the next one. That rhythm depends on two things working hand in hand: the muscle itself and the conduction system that tells it when to contract. Cardiac muscle combines the strength of skeletal muscle, which provides the power to pump, with the endurance of smooth muscle, which allows it to work tirelessly without fatigue. But power alone isn't enough. The heart has to beat in perfect coordination. That's how the concept of the functional syncytium comes in. Through microscopic connections called intercalated discs, neighboring cells are physically and electrically linked. These connections let ions, and therefore electrical impulses, pass quickly from one cell to the next. The result is that the entire myocardium behaves like a single unit. When one cell contracts, its neighbors follow instantly. So rather than millions of independent cells, think of the heart as one well-trained team, contracting together, relaxing together, and repeating that cycle for a lifetime. Now let's look closely at what makes the cardiac muscle to be designed for this non-stop job. At the microscopic level, cardiac muscles cells are striated, meaning they contain organized actin and myosin filaments arranged into sarcomeres. That's just like skeletal muscles. This arrangement gives the heart the strength and precision it needs to contract powerfully and rhythmically. But unlike the long cylindrical fibers of skeletal muscle, cardiac fibers are short, branched, and interconnected, each containing a single central nucleus. This branching pattern allows neighboring cells to form a strong three-dimensional network, giving the myocardium both strength and flexibility during each contraction. Each cardiac cell is also packed with mitochondria, occupying nearly a third of its volume. That's a clear sign that the heart relies almost entirely on aerobic metabolism. Can't afford to fatigue, not even for a moment. And finally, the cells are joined end to end by specialized structures that are both mechanical and electrical junctions called intercalated discs. Desmosomes within the discs keep the cells tightly bound, preventing them from pulling apart during powerful contractions. Gap junctions allow ions to pass directly between cells, enabling the rapid spread of electrical impulses. Together, these features make the heart a true functional syncytium, where many cells contracting as one coordinated unit. Let's now zoom in on one of the most defining features of cardiac muscle, the intercalated discs. If you ever wondered how millions of individual heart cells manage to contract as one perfectly timed unit, the answer lies right here. Each intercalated disc forms a strong connection between neighboring cardiac cells. Within the discs, there are two key structures working together: desmosomes and gap junctions, each with a unique but complementary role. Desmosomes anchor cells together firmly, especially during powerful contractions. Without them, the cells would literally pull apart every time the heart beats. Gap junctions, on the other hand, act like tiny doorways between cells. They allow ions, mainly sodium and potassium, to pass freely from one cell to the next. This electrical connection means that when one cell depolarizes, the impulse spreads almost instantly to its neighbors. The result is rapid, synchronized activation of the entire cardiac muscle mass. Together, these two features, mechanical anchoring by desmosomes and electrical continuity through gap junctions, make the heart a functional syncytium. In physiology, that means the myocardium behaves as if it were one single cell, contracting in perfect unison. Exam tip: when describing cardiac muscle structure, always include this phrase. Intercalated discs contain desmosomes for mechanical strength and gap junctions for electrical coupling, allowing the heart to function as a coordinated syncytium. That single line tells the examiner you understand both structure and significance. Taking a look at this table, we're going to compare the cardiac versus the skeletal muscle. This table summarizes key structure and functional differences that are often tested, not just for recall, but for interpretation. Let's go through them one by one. First, the cell shape. Skeletal muscles are long and multinucleated, built for powerful, voluntary contractions. Cardiac fibers, on the other hand, are short, branched, and contain a single central nucleus designed for endurance and connectivity. This branching allows them to form an interwoven network that supports uniform contraction across the myocardium. Next, control. Skeletal muscle is voluntary, activated by motor neurons. Cardiac muscle, however, is involuntary and myogenic, meaning the impulse to contract arises from within the muscle itself, specifically from the pacemaker cells of the sinoatrial node. This automaticity is what keeps your heart beating even without conscious control. Now, action potential. In skeletal muscle, it's very short, about 1 to 2 milliseconds, allowing rapid repeated stimulation. In cardiac muscle, the action potential lasts 200 to 400 milliseconds, producing that characteristic plateau phase. That long duration ensures each contraction is complete before another begins. This ties directly into the refractory period. And that's a life-saving feature actually, because it prevents tetany or sustained contraction, which would stop the heart's pumping ability entirely. Finally, gap junctions. These are absent in skeletal muscle, meaning each fiber contracts independently. In cardiac muscle, gap junctions are abundant, connecting cells electrically through the intercalated discs. This transforms the heart into a functional syncytium, allowing impulses to spread rapidly and synchronously. Exam tip. When you get a question like compare cardiac and skeletal muscles, don't just list the differences, explain their physiological importance. For example, the long refractory period in cardiac muscle prevents tetany, ensuring rhythmic pumping essential for life. Let's explore what happens inside the single cardiac contractile cell during one heartbeat. The cardiac action potential. Unlike skeletal muscle, where the action potential is brief, the cardiac one lasts much longer, about 200 to 400 milliseconds, like we said earlier, because it needs to coordinate a full, powerful contraction with time for relaxation. Let's walk through the phases step by step. The phase 0, or phase 0, this is the depolarization. This is the rapid upstroke. When the stimulus arrives from a neighboring cell, voltage-gated sodium channels open, causing a fast sodium ion influx. The membrane potential quickly rises upwards toward plus 20 millivolts. This rapid depolarization triggers the contraction process. Phase 1, initial repolarization. Immediately after, some sodium channels open briefly, allowing a transient potassium ion efflux. This creates a small notch on the graph, a short dip before the plateau begins. Phase 2, the plateau phase. This is the hallmark of cardiac muscle. Here, slow L-type calcium channels open, allowing calcium ions to enter the cell while potassium ions continues to leave. The two opposing currents balance each other, creating a long, flat plateau. This phase maintains depolarization, ensuring that calcium can trigger contraction and that the heart cannot be re-stimulated too soon. Phase 3, repolarization. As the calcium channels close and more potassium ion channels open, potassium efflux increases, bringing the membrane potential back towards its resting level. Phase 4, resting potential. Finally, the cell returns to its stable resting potential, which is about minus 90 millivolts. The sodium potassium ATPase pump and other exchanges restore ionic balance, preparing the cell for the next depolarization. Now, here's what's clinically and conceptually important. The long plateau phase corresponds to the absolute refractory period, meaning the cell cannot be re-stimulated during this time. This prevents tetany and allows the heart to contract and relax rhythmically, ensuring proper filling between beats. So when you're answering a question about the cardiac action potential, don't just list the ions. Emphasize why each phase exists. The plateau isn't just a shape on a graph. It's the reason your heart can beat faithfully, one contraction at a time. Now, a very important exam tip when describing this graph, as the cardiac action potential graph, always connect the ions to their functional significance. For example, the plateau phase allows time for calcium entry and contraction, preventing premature excitation. This demonstrates understanding, not memorization. Now that we understand the cardiac action potential, let's look at one of its most important consequences, the refractory periods that perfect the heart. After each depolarization, there's a built-in pause, a period when the cardiac cell cannot immediately be re-excited. This pause isn't a flaw. It's a brilliant safety feature that keeps the heart's rhythm steady and coordinated. Let's break it down. First, we have the absolute refractory period, which lasts about 200-250 milliseconds, roughly the duration of the plateau phase. During this time, most sodium channels are inactivated, so no new action potential can be triggered, no matter how strong the stimulus. This ensures that once a contraction begins, it runs to completion before another one can start. Then comes the relative refractory period, a brief period where some sodium channels recover, but the membrane is still hyperpolarized. Here, a stronger than normal stimulus might trigger a weak or uncoordinated contraction, but under normal conditions, This doesn't happen because the heart is protected by its timing system. Together, these two periods form the heart's electrical safety net. They prevent tetany and reduce the risk of arrhythmia, ensuring that each beat is followed by a full relaxation and filling phase before the next one begins. An exam tip: we asked to explain why cardiac muscle cannot undergo tetany. Link it to the long refractory period overlapping with the contraction period. You can say, because the refractory period lasts almost as long as the contraction, cardiac muscle cannot be re-stimulated before it relaxes, ensuring rhythmic, effective pumping. Now let's shift from the contractile cells to the pacemaker cells. These cells give the heart one of the most remarkable properties: automaticity, or the ability to generate its own rhythmic impulses without any external stimulation. Pacemaker cells are found in the sinoatrial node, atrioventricular node, and the Purkinje fibers, but the SA node is the primary one, setting the pace for the entire heart. What makes these cells different? First, they don't have a stable resting membrane potential. Instead, their membrane potential slowly drifts upward during what we call phase 4, depolarization of the pacemaker potential. This gradual depolarization is driven mainly by a special funny current carried by slow sodium and T-type calcium ion influx along with reduced potassium ion efflux. Once the threshold is reached, phase O depolarization occurs. But unlike contractile cells, this upstroke is caused by calcium influx through L-type channels, not sodium. This is why pacemaker action potentials are slower and less steep than those of ventricular muscle, but they are perfectly suited for their rhythmic role. Then during phase 3 repolarization, potassium channels open, allowing potassium to leave the cell, bringing the potential back down so the cycle can restart. This continuous cycle, depolarize, repolarize, drift, repeat, is what makes the heartbeat automatically around 60 to 100 times per minute at rest. For an exam tip, if asked about the ionic basis of pacemaker activity, always emphasize that pacemaker cells lack a stable resting potential and that calcium ion, not sodium ion, causes depolarization. That distinction often appears in multiple choice and short answer questions. So in simple terms, while contractile cells wait to be stimulated, pacemaker cells are self-starters. There are three main pacemaker regions in the heart, each with its own intrinsic firing rate or natural rhythm. At the top is the sinoatrial node, firing at about 60 to 100 beats per minute. It's the dominant pacemaker, the one that normally sets the rhythm for the entire heart. Because it depolarizes the fastest, it fires before any other region has time to reach threshold. Next in line is the AV node, which is a slower rate of 40 to 60 beats per minute. It acts as the backup pacemaker if the SA node fails. The AV node also introduces a slight conduction delay, allowing the atria to empty before the ventricles contract. Finally, the Purkinje fibers, firing at just about 20 to 30 beats per minute. They are the emergency pacemakers, slow but capable of keeping the ventricles contracting if higher centers stop functioning. This setup gives the heart a fail-safe system, multiple pacemakers arranged in layers of control. And here's the key point. The fastest pacemaker suppresses all the others, a concept known as overdrive suppression. In other words, the SA node keeps the slower ones quiet by continuously depolarizing them before they can reach threshold. But if the SA node is damaged, the AV node or Purkinje system can step in, though at a slower rhythm, which is why heart block or nodal failure often leads to bradycardia. An exam tip: if asked about pacemaker hierarchy, always mention the rates in order: SA, AV, Purkinje, and add that the fastest pacemaker dominates under normal conditions. Now let's put the pieces together and trace how a single electrical impulse spreads through the entire heart. This pathway is the reason your heart contracts in perfect order. Atria first, then ventricles, ensuring efficient blood flow every time. It all begins at the SA node, the heart's natural pacemaker, which is located in the right atrium near the superior vena cava. Here, spontaneous depolarization triggers an impulse that spreads rapidly through the atrial walls, producing atrial depolarization. This event is presented by the P wave on the ECG. This causes both atria to contract almost simultaneously, pushing blood into the ventricles. Next, the impulse reaches the atrioventricular node, where conduction slows down by about 0.09 seconds. This brief AV nodal delay is vital because it allows the ventricle to feel, allows it time to feel completely before they contract. Think of it as a well-timed pause that keeps the rhythm efficient. From there, the impulses travel into the bundle of His, which quickly divides into the right and left bundle branches, carrying the signal down the intraventricular septum. Finally, the depolarization wave spreads through the Purkinje fibers, which branch extensively into the ventricular walls. These fibers conduct impulses extremely fast, ensuring that ventricles contract from apex to base. That direction of contraction squeezes blood upward towards the seminal valves and out of the heart. The heart is empty. When describing the conduction pathway, always mention both the structures and the timing. For example, impulse spreads from SA node to the AV node, roughly 0.09 seconds delay in the AV node, then to the bundle of His, bundle branches, the Purkinje fibers, and then ventricular depolarization from the apex to the base. That level of clarity and sequence is exactly what earns full marks in physiology essays or viva. So how do electrical signals actually spread through the heart? Let's take a look. Not just where they go, but how fast they move. The cardiac conduction system works because every cardiac cell is electrically connected to its neighbor through gap junctions located in the intercalated discs. When one cell depolarizes, positive ions, mainly sodium and calcium ions, flow directly into the next cell, causing it to depolarize as well. This process allows the impulse to jump rapidly from cell to cell, creating a wave of excitation that travels through the heart's muscle like a coordinated ripple. However, the speed of conduction isn't uniform. It varies across different regions of the heart, and this variation is what allows the chambers to contract in perfect sequence. Let's look at the key differences. The AV node conducts slowly, about 0.05 meters per second. That slow speed is deliberate. It creates the AV nodal delay, giving the ventricles time to fill with blood before they contract. The Purkinje fibers, on the other hand, are the fastest, conducting at about 4 meters per second. This rapid conduction ensures that the ventricles depolarize almost simultaneously from apex to base, producing a strong, coordinated contraction. The rest of the myocardium, that's the atrium and the ventricular muscle fibers, conducts at intermediate speeds, balancing coordination and efficiency. The key takeaway here is that the variability in conduction speed is not a flaw, it's a timing mechanism. Slow, where precision is needed, that's the AV node. Fast, where power and synchronization matter, Purkinje system. When you are writing about the conduction system, don't just mention where the impulse travels. Explain why it travels at different speeds. So let's pause for a moment and connect everything we've learned so far. We talked about how the heart generates and conducts electrical impulses, but how do those impulses actually produce the heartbeat we can feel? That's where excitation contraction coupling, or EC coupling, comes in. In simple terms, EC coupling describes how an electrical event, in this case the action potential, is converted into mechanical event. That's the contraction of the cardiac muscle. The bridge between these two walls is calcium. When the cardiac cell depolarizes, calcium enters the cell, triggering the release of even more calcium from internal stores. That surge in intracellular calcium allows the contractile filaments, actin and myosin to interact and generate force. An exam insight. If a question asks, explain excitation contraction coupling in cardiac muscle, the opening sentence should always make this clear. Excitation contraction coupling is the process by which electrical depolarization of the cardiac membrane leads to a rise in intracellular calcium and subsequent muscle contraction. That single line sets the tone for a full, well-organized answer. Now, so here we are looking at the complete process of excitation contraction coupling, the vital link between the heart's electrical signals and its mechanical contraction. Let's trace the sequence carefully from left to right. When an action potential travels down the cardiac muscle fiber, it spreads into the T-tubule. Alright, shown on the left side of the diagram. This depolarization opens the L-type calcium channels, also called dihydropyridine receptors. These channels allow a small but crucial amount of calcium to enter the cytoplasm from the extracellular space. This incoming calcium doesn't cause contraction directly. Instead, it acts as a trigger. It binds to and activates the ryanodine receptors on the sarcoplasmic reticulum. Through this mechanism called calcium-induced calcium release, a much larger amount of calcium is released from the sarcoplasmic reticulum into the cytosol. Now, inside the cell, calcium quickly binds to troponin C on the thin filaments of the sarcomere, shifting the tropomyosin strands away from the myosin binding sites on actin. This exposure allows cross-bridge cycling between actin and myosin, generating tension and causing the heart muscle to contract. Now, contraction can't last indefinitely. For relaxation to occur, calcium must be removed from the cytoplasm. This happens mainly through three pathways. The Sarco pump, that's the sarcoendoplasmic reticulum calcium ATPase pump, which actively pumps calcium ion back into the sarcoplasmic reticulum, and this is regulated by phospholamban. Second, the sodium calcium exchanger on the cell membrane, considered here. This swaps one calcium ion for 3 sodium ion. 3. The sarcolemma calcium ATPase, which directly pumps a smaller amount of calcium out using ATP. Now, the sodium potassium ATPase maintains the ionic gradients that keep this exchange system running, while mitochondria is nearby to recycle ATPase, which fuels these pumps. Now, once the cytoplasmic calcium level falls, troponin releases calcium, cross bridges detach, and the sarcomere relaxes, completing one full cycle of excitation, contraction, and relaxation. Now, finally, notice the intercalated disc on the right, containing desmosomes, for mechanical linkage and gap junctions, for electrical continuity, ensuring the excitation spreads smoothly from one cell to the next. So the entire myocardium contracts as one coordinated unit. This is a very long one. So here you have outlines of excitation, contraction, coupling in the cardiac muscle, the steps involved, depolarization, calcium-induced calcium release, cross-bridge activation, relaxation, coordinated response. Now the exam tip, always mention calcium-induced calcium release and circa when describing cardiac excitation contraction coupling. They are the defining features that distinguish it from skeletal muscle. Now that we've seen how calcium drives contraction, let's bring this into real-world context as how drugs can modify these same processes to treat heart disease. Every one of these medications works by adjusting calcium availability or handling within the cardiac muscle cell. Let's start with the calcium channel blockers. Now these drugs like verapamil and diltiazem block the L-type calcium channels that normally allow trigger calcium to enter during the action potential. Less calcium entry means less calcium-induced calcium release from the sarcoplasmic reticulum and therefore reduce contractility. Clinically, this helps to lower the heart's workload and oxygen demand. This is useful in conditions like angina and hypertension. Next, you have a drug, digoxin, that's a cardiac glycoside, which works in the opposite direction. It inhibits the sodium-potassium ATPase, which indirectly reduces the activity of the sodium-calcium ion exchanger. As a result, more calcium stays inside the cell, increasing the calcium available for each contraction, producing a stronger heartbeat. That's why digoxin is used to improve cardiac outputs in heart failure. Finally, beta blockers, such as metoprolol or propranolol. These have a more long-term effect. By blocking sympathetic stimulation, they reduce heart rate and calcium overload, allowing the heart to relax more fully between beats. Over time, this improves calcium handling efficiency and reduces arrhythmias, leading to better mechanical performance with less stress. The key takeaways here is that all of these drugs, whether they block, enhance, or fine-tune calcium movement, all work by modifying the same excitation contraction coupling pathway you've just learned. And that's why understanding calcium physiology is foundation for understanding nearly all cardiac pharmacology. So remember, in the clinic, when we give these drugs, we're not just changing numbers on a monitor, we are modulating the rhythm and strength of life itself, one calcium at a time. A moment to appreciate what the electrocardiogram really shows us. It's often described as the electrical window to the heart because it lets us see what's happening electrically inside the myocardium from the outside of the body. The ECG doesn't measure the activity of a single cell. It records the summed electrical currents generated as millions of cardiac cells depolarize and repolarize in a coordinated sequence. In other words, every wave you see on the surface is a reflection of the heart's internal choreography of ions moving across membranes. It's also important to remember that the ECG is an extracellular measurement. Now let's decode the key landmarks you'll always see on a standard ECG. The P wave represents atrial depolarization. The QRS complex reflects ventricular depolarization. The T wave shows ventricular repolarization. Now these together, these three deflections of the P, QRS, and T form the rhythm of every heartbeat. Electrical excitation, contraction, recovery, readiness for the next cycle. Now a small exam tip, if asked, what does the ECG represent? The first line should be this. The ECG records the summed extracellular electrical activity of the heart during depolarization and repolarization, represented by the P wave, the QRS complex, and the T wave. Now that we understand the heart's intrinsic rhythm, let's see how it's finely tuned by the autonomic nervous system. That's the body's built-in regulator that decides when the heart should slow down, speed up, or pump harder. At rest, both parasympathetic and sympathetic nerves are active, but the vagus nerve usually dominates. This parasympathetic influence releases acetylcholine, which binds to M2 receptors in the SA and AV nodes. The result is a slower heart rate, what we call a negative chronotropic effect. When the body needs to respond, say during exercise, stress or excitement, the sympathetic system takes the lead. Here, norepinephrine acts on beta-1 receptors in both nodal and ventricular tissue. This causes three key effects: a faster heart rate, known as positive chronotropy, a stronger contraction, also positive ionotropy, and lastly, faster conduction through the AV node, positive dromotropy, which also shortens the AV delay, so the atria and ventricles stay perfectly in rhythm. This rapid, coordinated response boosts cardiac output and ensures blood and oxygen delivery rise instantly when the body demands it. Now, what's fascinating is how the balance between these two systems shifts throughout the day. At rest, vagal tone predominates, keeping the heart calm and energy efficient. During exercise, sympathetic drive surges, the accelerator takes over, increasing rate and force. Clinically, you can manipulate this balance with drugs. Beta blockers dampen sympathetic activity, lowering heart rate and contractility. This is helpful in hypertension and arrhythmias. Atropine blocks the vagus nerve, allowing the SA node to speed up, and is used to treat bradycardia. Digoxin enhances vagal tone while increasing intracellular calcium for stronger contractions, and is valuable in heart failure and atrial fibrillation. So here's the key idea. The heart is always under a dual command. The vagus as the brake and the sympathetic nerves as the accelerator. And most drugs simply shift that balance to meet clinical needs. In exams, when you're asked about autonomic control, always describe both systems, their receptors, their effects, and one clinical example. That shows you understand the physiology and its therapeutic relevance. Here's a quick look at what happens when cardiac physiology breaks its rhythm. In myocardial infarction, blood flow stops, cells starve, and CT elevation marks electrical injury. Every second counts because time is muscle. Atrial fibrillation turns the atria chaotic, hundreds of impulses, no P waves, regular beats, and poor feeling that invites clots. In complete AV block, the atria and ventricles lose communication. Two rhythms run separately, and only a pacemaker can restore order. Hyperkalemia raises extracellular potassium, dulling the resting potential. The T waves peak, QRS widens, and danger looms for cardiac arrest. Finally, calcium imbalance. Too little prolongs repolarization and triggers arrhythmia. Too much shortens it, tightening the beats, but risking instability. Different causes, one message. When ions, oxygen, or conduction falter, the heartbeat's perfect rhythm begins to fracture. Before we wrap up this topic, let's clear up some few common misconceptions that often trip students up in exams and vivas. These points may look simple, but they separate rote learning from real understanding. 1. ECG, individual action potentials. The ECG does not record the electrical activity of one cell. Instead, it represents the summed electrical currents produced by millions of cardiac cells as they depolarize and repolarize together. So when you see a P wave or a QRS complex, think of it as the collective activity of the whole heart, not a single fiber firing. 2. Calcium comes from both the extracellular fluid and the sarcoplasmic reticulum. About 20% and 8% respectively. That small influx of extracellular calcium ion through L-type channels is what triggers the larger release from the SR, the process called calcium-induced calcium release. Without that 20% trigger, contraction will be weak or absent. 3. The SA node initiates the impulse. It doesn't contract. This is a very common mix-up. The SA node is a cluster of specialized pacemaker cells that generate rhythmic action potentials, but they are not contractile. They tell the rest of the heart when to beat. They don't contribute to the pumping action. 4. The plateau phase of the action potential is not a rest period. Even though the voltage looks flat, the cell is in a state of intense ionic exchange. Calcium is entering while potassium is leaving. It's an active phase that maintains depolarization and sustains contraction. So don't call it a pause. It's the heart's power hold moment that keeps contraction coordinated. Exam tip: if a question tests conceptual accuracy, sure you understand these nuances. For example, the ECG reflects the sum of cardiac potentials, not individual ones. Or the plateau phase sustains contraction, preventing premature excitation. These short, precise clarifications make your answers sound confident and expert. So as you revise, remember, physiology isn't about memorizing the facts. You'll be repeating it these over and over again. It's about understanding mechanisms and knowing what's really happening beneath each wave and ion channel. To bring this topic to a close, let's take a moment to step back and see the big picture. How everything you've learned about the cardiac muscle and conduction system fits together in one elegant, coordinated process. First, remember the foundation. Intercalated discs connect cardiac cells both mechanically and electrically. Through desmosomes and gap junctions, they allow the heart to function as a syncytium. That's many cells contracting together as one. That's the secret behind the heart's seamless rhythm and endurance. Next, the long refractory period. This is the heart's built-in safety feature. It ensures that each contraction is followed by complete relaxation before the next one begins, preventing tetany and keeping the rhythm clean and efficient. Then think of the conduction sequence. The perfectly timed relay that starts in the SA node, pauses at the AV node, and spreads through the bundle of His and Purkinje fibers. This system ensures that depolarization and contraction happen in a coordinated way. Atria first, then ventricles, pumping blood smoothly through the circulation. Next, we have calcium, which is the star player linking electricity to muscle movement. Every beat begins with an electrical impulse, but it's calcium that turns that impulse into mechanical force through the excitation contraction coupling. Without calcium, There's rhythm but no contraction. A heart that's alive electrically but motionless mechanically. Finally, the autonomic nervous system fine-tunes everything. Parasympathetic input via the vagus nerve slows the rhythm for rest and recovery, while sympathetic stimulation speeds and strengthens the heartbeat during stress or activity. Together, they maintain a perfect balance, adapting the heart's performance to every demand, from sleep to sprint. So what's learning without testing what you've actually gained? So here are a few selected questions to test your knowledge and understanding. These questions test your understanding of the topic, that's the cardiac muscle and the conduction system. So for those of you that have made it this long, here's a little bonus slide, and it's how to structure your answer. So whenever you see a question on cardiac muscle or conduction, don't start writing immediately. Pause for a few seconds and ask yourself, what exactly is this question testing? Is it about structure? Is it about conduction? Is it ionic mechanisms or functional comparison? Identifying the question type helps you decide what to emphasize and how to organize your thoughts. Begin with a focused introduction. Define the key concept. For example, cardiac muscle is a specialized striated muscle adapted for rhythmic, involuntary contraction that pumps blood throughout life. That one sentence already tells the examiner you know what you're talking about. Then move into your main body, which should flow logically. Start with structure, describe features like branching fibers, intercalated discs, and the syncytium. Then explain electrical events, the action potential phases, and the conduction pathway. Finally, connect both of them. Show how structure supports function and how electrical activity translates to mechanical contraction. Then, very importantly, don't forget your diagrams. Simple, clearly labeled drawing of the conduction pathway or cardiac cell structure can earn easy marks and make your answer stand out. Lastly, end with a short, purposeful conclusion. Summarize the physiological principle and, if possible, mention one clinical correlation. Like how disruption of the conduction system can lead to arrhythmia or heart block. And remember, in the exam, it's not about how much you know, it's about how clearly and confidently you can explain it. A well-structured, logically flowing answer shows the examiner that you understand both what happens and why it matters. As we conclude this tutorial on cardiac muscle and conduction system, I want you to take away more than just the facts. Remember that physiology is not about memorizing mechanisms. It's about understanding why they work together. The heart isn't simply a pump. It's a living rhythm, a union of structure, ions, and electrical precision that sustains every other system in the body. So as you prepare for your exams, think beyond definitions. Visualize how each cell contributes, how every delay or ion movement has purpose, and how structure serves function in every beat. Thank you for staying engaged and curious. Keep revising smartly, keep thinking deeply, and never forget that physiology is the study of life in motion. See you in the next tutorial. Until then, keep your heart and your mind in rhythm.